going to be doing from here on out for the rest of the semester is going through organ systems. So we've done all of our basic background kind of biology with the stuff, right, with the cells and the chemistry and so on, right? So now we want to start looking at the organ systems. And remember that the definition of an organ system is basically going to be a group of organs that are all functioning together to carry out a particular function. And so the first one we're going to be talking about is, of course, the integument, which means the skin as an organ system. And so the skin, and so now we're down here, of course, the equivalent of that right there. You know, again, in terms of our sections, our pictures, and our models in the laboratory, just basically three parts to it. The epidermis, the dermis, and the subcutaneous layer. Those are the tissues that are in the skin. And then otherwise, we have a variety of other organ-type structures like hair, like glands, like receptors, and things like that, that all together carry out the functions of the skin. So the first thing I'm going to do here is to list some of the functions of the skin itself. Kind of talk about them for a second. I will say that the skin itself, the integument, is the largest organ system. And so if you were to take me and skin me like a deer and lay my pelt out here on the floor, you would see it'd be about a couple meters squared in terms of surface area. Weighs about 9 to 10 pounds. You know, how much of this stuff you happen to be carrying around with you, you know. So it is physically the largest of the organ systems. By the way, you ever play that HQ trivia game? You ever heard of that? How am I with it? Have you heard of it? Yeah, anyway, actually one of the questions last week, what's the largest organ system? It is the skin, right? I don't believe that I'm doing something that nobody else except one person is doing it here on the internet. But anyway, every night, like 8 o'clock or something, it comes on. All right, so meanwhile, functions of the skin, of the integument. So, you know, obviously we do have protection, right? And so, thing is that we've got a physical barrier. This is particularly because of the stratified squamous epithelium. You know, it's real thick in terms of the epithelial layer there in the epidermis. Another important function is that it does regulate your body temperature. And there are 3 or 4 different ways that this is going to occur, but sweat glands, of course, are one of the ways in which we do cool the body by putting perspiration out onto the skin. Another important mechanism just has to do with blood flow to the skin. So you can increase or decrease the blood flow to the skin surface. When you increase blood flow to the surface, that brings more heat from the interior out. It can radiate out at the surface. If you diminish blood flow to the surface, holds more heat on the inside. Okay? Another thing is we do have a number of receptors out in the skin. So things for temperature, for instance, touch, pain, pressure. Okay. Detecting again what's happening at the outside. This is basically the way your brain figures out what's happening actually out on the surface of the body. We have excretion as a function. This gets back to basically the sweat glands again. So not only are you cooling, but you're also eliminating excess water. And also certain kinds of solutes from the body. So you know when you sweat a lot, you feel all kind of like grainy out here. anyway, solutes coming out as well. We have immunity as a function. All right, and so earlier I was saying that with connective tissue, you've got a lot of immune cells, and so that is the case right here in the dermis. We have one particular type of immune cell called a Langerhans cell that you'll see in the epidermis. Okay, that just sort of is a physically stationary macrophage in the epidermis. You'll see it, but it's there again to kind of catch anything that's coming through the epidermis at that particular location. So you'll see that on one of my pictures, the pictures here. All right, the next one right here, kind of weird. It does turn out that the synthesis of vitamin D actually starts in the skin and the initial stage. And so we have as a precursor here to vitamin D, cholesterol. And cholesterol is going to be converted to the vitamin D precursor in the skin. And it's going to do so in the presence of UV light. UV basically provides the energy for this first step to occur. I'll show you a picture of that in a second. After you get the vitamin D precursor, goes off to the liver and then ends up at the kidney. Basically, vitamin D is going to help what? Your body would absorb calcium from your digestive tract. That's the main function for it. And then otherwise, in general, it prevents water loss. Cushions the body. Also insulates the body. Okay, so these are some functions of the integument. All right, and then since I guess I should say no articles again saying that we do need some degree of UV light right here in order to actually convert cholesterol to vitamin D. The estimate it takes about 15 minutes a day of sunlight exposure for this to happen. But as you also know, we've got problems with skin cancer related to UV light. So let me actually back up over here under the protection part and also mention that we will have this pigment melanin in the skin that's going to basically protect the skin from UV light. Again, skin cancer is basically directly related to exposure to UV. So we got a bit of a contradiction here. We need some for the vitamin D part of it, but too much is a problem. And again, 15 minutes per day of UV kind of exposure, that's kind of average throughout the country. Here on a regular old day, we probably need what? 5 minutes? Because we get excess, right, But where you go further north, where it's not quite as bright sun and so on, then a bit more time per day. All right, so anybody got a thought on my functions here? All right, here's the deal about the blood flow. We'll talk about this again later on. We're just talking about the circulatory system. But you're running, you're sweating. You can see the sweat glands are pumping fluid out. Body internal core heating up, right? All this metabolism going on. And so one way to get rid of excess heat is just to increase the blood flow here to the skin. Notice the blood vessels are in the dermis, they don't go into the epidermis. Again, that's characteristic for epithelium, but that radiates the heat, excess heat, out of the body, helps to keep the body cool. This guy over here standing up on the North Pole, right, of course, not really ultra sweating up there, so sweat glands probably hardly even ever function up there. But the thing is because he's more concerned with conserving body heat, then that means that as far as the blood flow goes, you decrease the blood flow up here. Still a little hint of heat radiating out, but you're actually conserving most of it down in the body. And again, this is a kind of decreased flow versus increased flow above normal or below normal, right? There's always some degree of flow going there. But again, that's a way to, again, keep the body temperature regulated. Again, just showing the same thing. We'll talk about this term, vasodilation, which just means the capillary just what? Big around as possible, lets the most blood flow through it. So we are vasodilating there. You can see the bulk of the flow is coming to the skin rather than following the main conduit. So you radiate the heat, but over here on this side where you want to conserve body heat, most of the flow comes this way, just a little bit that direction there. Okay, here's the thing about vitamin D. So again, what we're talking about is up here at this point. So there's cholesterol, there's the UV light at the surface, being converted cholesterol to this precursor deal. Ends up in the liver, ends up in the kidney, and then you've got your functional vitamin D. And again, what it does is it helps to increase the absorption of calcium and phosphate, basically, from your diet. Particularly calcium, that's our main emphasis right here. And then calcium used in a lot of ways, including building your skeleton. All right, so that's that. Now, the thing I always like to show is the thing about the skin, as you probably know, like, you know, like this, right? A bunch of layers to it. You know, when they say beauty's only skin deep, they're not kidding. Because once you look under that, we all look pretty funky, right? Down there at the muscle layer. So you can see off on the edge muscles, but you can also see a very precise dissection of the cadaver skin. So we have here on the outside the epidermis itself, and so here we have our surface right there, and again it's ultra thin. Here is the dermis, which is going to be 3 times, say, on average, thicker or more, probably thicker than the epidermis. Here is the subcutaneous layer, the fat layer. It is kind of interesting if you ever hurt yourself to the point where you go through the epidermis, through the dermis, and you're down in the fat layer, you will see these little yellow blobs of tissue. That is the color that it is. And then we have under the subcutaneous layer this connective tissue band called fascia, and then it's over the muscle itself. And again, the epidermis, depending on where you're looking at in the body, is going to be kind of sort of uniformly thin, but again, thicker right here, for instance. Dermis also kind of has a uniform thickness to it, but it is a little thicker right here. And then the subcutaneous layer itself, that is unfortunately the layer that kind of varies, right, over the course of your life, because, you know, that's the one that actually can get a little bigger, a little thicker, thinner, right, according to what you're doing in life. So, cadaver dissection, and again, it's a very precise dissection. And so again, those 3 layers that's the skin right there. All right, now, as we start looking at this then, you know, the epidermis first. So it is thin, but it's multi-layered. It does have stratified squamous epithelium, which means when you look here and go up to the surface up there, lots and lots of cell layers. The squamous means that the layers on the surface are real thin and flat, and they're actually dead at maturity. They're going to always kind of flaking off. It's avascular, which means there's no blood vessels. And they come up, as you can see right here, just to below the epidermis, in the dermis, but they don't actually physically extend into the epidermis itself. Now, as we think about the structure, again, here's the picture I showed you in the laboratory for the micrograph, and also the one you need to look at for the slide if you haven't. Again, the epidermis is here to the top. Here's our dermis with the dense fibrous, and then here is the adipose tissue down here for the subcutaneous layer. Now, we want to sort of understand the relationship of the layers of the epidermis to each other. So let's think about that right now. There are 4 layers to the epidermis in regular skin, often called cornified. But in the palms of your hands and the soles of your feet, you do have one additional 5th layer. So in lab, we were talking about this, and so I kind of drew the picture up there and then. We start out down here at the boundary between the epidermis going this direction and the dermis going this direction here with a single layer of cells. And this single layer of cells is called the stratum basale. Okay, and so that's just that single layer of cells there. We have a layer of cells up more toward the surface or superficial, a layer of about 4 to 6 or so cell layers. That is called the stratum granulosum, always stained fairly distinctively dark relative to the layers above and below it. Losum. All of this tissue here between the stratum basale and the stratum granulosum is called the stratum spinosum. Okay, so basically from the top of the basal layer to the bottom of the granulosum layer. And then we have out toward the surface our most exterior layer, and this is called the stratum corneum. So those are the layers that are in all skin, okay? Now, in the palms of your hands and the soles of your feet, one additional layer, and that layer forms kind of right along the kind of surface side of the stratum granulosum. This is called the stratum lucidum.
Thank you.
Palms of your hands and the soles of your feet. So that's the layer again that's present in again this stuff here on your hands and on your feet, which of course is just inherently thicker skin structure. Now, as you look at a micrograph, of course we're doing this on the cell model in the laboratory, but as you look at the micrograph, the dermis is always light stained, the epidermis is more darkly stained. So the little layer of cells just kind of follows the contour of the dermis as the stratum basale. Here is the granulosum layer itself, which always stains darker, about 4, 5, 6 layers or so. And then these cells here between the basal layer and the granulosum, stratum spinosum. And then everything from the granulosum all the way up to the surface itself, surface of the skin, is the stratum corneum. And that's the part that shows that flaky, thin, squamous sort of cell effect for the stratified squamous characterization for the tissue. And again, lots of cell layers. Again, as you get to the top of there, thin, flat cells. All right, now the basal layer, deepest, which means it's the furthest from the surface next to the dermis. Single layer of kind of cuboidal shaped cells. Okay, so on here it's this bound layer of cells right here. Here's the dermis at that point there. Melanocyte cell bodies are located here within the stratum basale. And so we're showing right here 2 melanocytes. These produce that dark pigment melanin, and the function of that is to protect the skin and also the hair and also the iris of your eyes from ultraviolet radiation. And so the melanocytes themselves, again, cell body in the basal layer, but they have little extensions that kind of grow up into the upper layers as well as down below a little bit. And then they deposit the pigment, these little black deals. You can see little black marks right there represent the melanin pigment. It always gets deposited on the surface side of the nuclei here for the skin cells. That's how we're kind of protecting the nuclei from the effect of the UV coming in this direction right here. The other thing about the stratum bassalis is that it is capable of cell division. And again, that is a characteristic of all epithelium. Because it's at the surface, it's going to get damaged, right? So you've got to replace the cells that are damaged, otherwise it's going to get, thinner and thinner. And the thing about our skin is the surface always wearing off. It's always kind of flaking off, and it's supposed to because it's dead. That's its functional kind of characteristic. But as you're flaking off here, if you don't have a corresponding, production of cells from the bottom, that means over time it's going to get thinner and thinner. So they're showing a little cell division kind of metaphase stage right there to indicate cell division has occurred. Every single cell here capable of cell division. And that's an unusual characteristic of cell division, particularly when you get to be an adult. Okay? So that's two important characteristics for the stratum bassalis. Melanocyte cell bodies are located there, and then it is capable of cell division. Now, the stratum spinosum is going to be these cells up through here. Now, the cells that are found here in the first set of layers above the basali layer are alive. The functional cell type for the epidermis is called a keratinocyte. So that's the functional epidermis cell. But they're going to actually form and ultimately, end the spinosum layer. But then they ultimately will die when they get to the granulosum, because they're only functional and when they're dead. So as we kind of look at the illustration here, you can see that the cells are still kind of cuboidal shaped as we're here, but as they continue to add more cells, they get pushed up this direction. And then ultimately they develop into a keratinocyte. And that's the functional skin cell for the epidermis, produces this material called keratin, which is ultimately going to waterproof and provide a degree of antibacterial activity to these cells, but it's going to kill them. It's going to kill them. And so as we start to get up to this point right here, the keratinocytes, may already be dead, but if they're not dead at this point, when they go through the granulosum layer, they will be dead. And then once they come out on the other side of the corneum, now they are really functional epidermis cells. Here is that reference to the Langerhans cell I was mentioning earlier. It is an immune type cell. It's a macrophage type, but it's a type of macrophage. Some of them kind of wander around in the body. Some of them are kind of fixed. This is one of these that are fixed. You'll see it later when we talk about immunity. But its basic function is to sit here in this location in the skin. She's got all these little extensions out here. So if damage happens right there, it's right there where something would come in from the outside into the skin. And again, the idea is trying to intercept stuff here before it gets down deeper into the skin. So immune function. Again, the granulosum always stains darker than the spinosum and the cornea, typically stippled looking. But again, as a cell comes into this side of it right here, if it's not dead at this point, when it comes out on the other side, it is dead. Okay? And then this is not illustrating the lucidum, which would be here. So again, when we come out of the granulosum right into the cornea. And again, waterproofs, protects from injury, and also microbes. And then the corneum itself is the thickest part of the epidermis. It's up at the surface, so from here to wherever the top is up there. And again, all those cells are dead, and they're always flaking off. Always flaking off. And again, as they flake off here, you get a corresponding rate of cell division down here to continually make however many cells you need to maintain the thickness of your corneum layer. All right, so everybody see that? Everybody see that? Anybody not? Particularly important, kind of recognizing the sequence, deep to superficial, Because again, in the lab, I do ask you on that one plaque model to be able to identify the layers of the epidermis. Everybody okay with this? All right, so again, just showing the same thing. Stratum basale, showing a melanocyte right there, melanocyte there. Again, little extensions go off between the cells. They deposit the melanin. These are the Langerhans cells right there. And then again, we have our spinosum layer, granulosum layer, again showing the lucidum here for the palm, and then everything above that, stratum corneum. Okay. All right, again, more about the melanocytes, pretty important cells actually, and so again, they deposit melanin. And the thing about it is it mostly ends up on the surface side of the nuclei. UV's coming in this direction right here, so you want all your protection on this side so that it won't be damaged by the UV. All right, and you shouldn't do this one. All right, so again, anybody got a thought here on that? Here's the picture I show you for, and we look at the slide as well in lab just for tissue, stratified squamous epithelium. So again, here's the dermis here. So again, the basal layer, spinosum, granulosum, corneum up there to the all right, and then otherwise, palm skin versus other skin. Our skin right here kind of has sort of a uniform thickness to it. You go around the body, 4 layers. Thinnest skin's up here, eyelid, right? Still got all 4 layers, but they're ultra thin, individually. But of course, when you do go right here, this is where all your effort goes into stuff, right? And on the bottom of your feet, things are thicker. So this is just illustrating that with the so-called thick skin, which means palm, for instance, that we do have a significantly thicker overall epidermis, particularly at the corneum part of it. But on this one, that little band of tissue, kind of orangish, represents the lucidum layer. Dermis itself, not necessarily so much thicker, but you got a lot of fiber right there in the dermis. And then as far as the thin skin, like on your arm, for instance, you can see that the corneum isn't particularly thick relative to what we have over here on the palms of your hand, soles of your feet. All right, so keep saying the same thing. Anybody got a thought on this? And by the way, in case you hadn't realized, you can say, I don't get it. It's when nobody says anything that I'm scratching my head. So anyhow.
I have a question.
Go ahead.
What was the extra layer in the skin?
The lucidum.
Yes.
In the
Are
palm.
you born with that, or?
Yeah.
Because I know babies are super soft and you don't really.
Yeah, but that's just because you haven't, the babies haven't developed quite as much fiber, haven't developed quite as much, that keratin and fat.
Yeah.
They're supple. Yeah. No, they've got them all. It's just they're much less developed than they are as they get to be what? Child, teenager, adult. All right, so with that, let's let's talk about the dermis. All right, so dermis got one kind of tissue. It's that dense connective tissue, lots of fibers. All right, so thing about the dermis itself, just split up into 2 parts. So it extends from the bottom of the epidermis, which is our stratum basale, all the way down to the top here of our subcutaneous layer. We've got a little section right here in the upper dermis, which is called the papillary layer. And then everything basically from here all the way down to the bottom is called the reticular layer. And that's the biggest, thickest part. Now, the papillary region itself, papillary layer, is characterized by the so-called dermal papilla, which are these little kind of extensions and kind of protuberance that you see here at the top of the dermis. And so you don't have actually a flat surface boundary between the dermis and the stratum basale. It's all kind of highly irregular. And what that papillary business does, dermal papilla does, is it first of all increases the surface area up here at this point, for basically moving stuff from the blood vessels into the epidermis. And the blood capillaries are always up here. I'm only showing one, but they're in each one of these. And that's where you actually get your stuff moved into the epidermis. No blood there, so you don't have any direct delivery, so you have to depend on kind of diffusion of materials. The more surface area you have, the more opportunity for diffusion of materials into the epidermis. The other issue related to the papillary layer is that they also provide your fingerprint type of patterning that everybody has. It's pretty characteristic for each person. And so all of us have a unique way in which these papilla are organized relative to each other. We see them kind of just cut in a section right here, but they actually kind of follow in kind of rows and bands and all kind of weird kind of, just kind of complicated kind of arrangements. Okay? So anyway, the papillary, again, dermal papillae, increase the surface area, form fingerprint patterns, and again allow just for more diffusion per area than just having a very straight, flat boundary right there. And then the other thing that we have is the receptors. And so we're showing right here a Meissner's corpuscle. Meissner's corpuscles for a kind of structure are super light touch detectors. We mostly see these in like our fingertips. That's where most of the time we're kind of sensing the environment, right? With your fingertips. And so the Meissner's corpuscle is one of these kind of oval white looking structures, and you can see one right here, kind of found up in the kind of dermal papilla uplifted area. And so again, receptors for very light touch. On the models in the lab, again, you want to look at the dermal papilla, and some of them have the little Meissner's in them, but you pretty much have them everywhere on your fingertips. Rest of the skin a little bit less, less concentrated. All right, and then otherwise, as we come down here toward the base, lower part, not the base, the reticular layer, again, from about right here all the way down to here, that's, again, our connective tissue. We've got Pacinian corpuscles, we've got hot and cold temperature receptors, and then we have our other types of hair follicle, other glands, and so on. And so on the picture right here, we're illustrating a Pacinian corpuscle right at this location here. They can be in the lower dermis reticular layer, or they can be in some cases in the subcutaneous, just depends. But these are more directly sustained pressure, heavy pressure types of receptors. So anything that's real delicate and light, your Meissner's will detect. But if you're really kind of pushing here on the skin, then you're actually pushing down a little bit below. That's going to activate these Pacinian corpuscles, also called lamellated corpuscles. We have, in addition, the hot and cold temperature receptors. For something that's basically responding to temperature, they're usually just kind of free nerve endings. So you can kind of see some right there, see some right there. They just come up and the ends just kind of respond to temperature there at the surface. And then the accessory structures themselves, which include the hair follicles, sebaceous glands, and the sweat glands, eccrine glands, as well as the glands here in our ear canal. Ceruminous gland, also located in the dermis as well. And then we'll come back and talk about those in a second. All right, so bulk of our receptors then here in the dermis. And then otherwise, subcutaneous layer, which is also called hypodermis, because hypo means below the dermis, again, is the adipose tissue, and essentially what secures the dermis here down to the underlying muscle down below the giddy. All right, so this is illustrating the dermal papilla. So you can see we're showing two of those right there, and again, just way more surface area for movement of materials. Again, the way they kind of arranged here in terms of your own self, they generate the fingerprint patterns. We don't really talk about the main tissue difference between papillary layer and reticular, but there is, and it's still connective tissue and fibrous connective tissue. It's just a little less intense in the papillary region. Again, just illustrating again on the micrograph, that's the dermal papilla, and so on. Here's the thing about how they do form the fingerprint layers. And so, we see them cut in section like this, but every time you see a section like this, it actually is extending, of course, back into the view as a ridge, right, as a ridge. And again, it takes on its own characteristic patterns that, of course, unique for us, pretty unique, fairly unique, significantly unique for us as individuals. But again, provide kind of uplift there. Illustrating the Meissner's corpuscle at the top. Again, up at the surface where very light, delicate touching, soft touching would be detected. These will not detect anything that's real light and delicate, but if you start pushing down here onto the surface of the skin, then these will respond. Those will basically over-respond right there. These will respond. This is just illustrating that the Pacinian corpuscle itself looks like an onion in section when you do look at it. And so the nerve ending in the center, and then you got all these like little layers of nerve tissue around it that basically, again, respond to the kind of force of the touch. All right, so that's that part. All right, so for the last few minutes, let's say a word about the hair and the exocrine glands and nails if we get there. So as we do, of course, think about the skin. We've got hair pretty much everywhere on the body. Only a couple of places that we don't really have any hair. The palms and soles of your feet are one. Your lips, you got hairy lips. Probably not, right, Tips of your fingers, toes. All the same difference. But the areola sometimes has some hair, right? Sometimes, right? Yeah. But generally everywhere else, okay? And the hair itself is pretty important for, a variety of functions. So, protects organs, cushions organs, insulates, filters air. We've got a lot of hair here in our nose, for instance, and of course can't catch debris coming in. A lot of it in your ear canals as well. Shades your eyes, both with your brows a little bit, but your eyelashes, and eyelashes themselves act as receptors for anything that might fall down and hit it, cause your eyes to close, so it won't get in your eye. If you think hard, you can touch your eyelash, obviously, without making your eye blink, but if something just kind of hits it without you sort of thinking about it, it will cause your eye to close and reflex blink. Hair composed of dead cells, too, just like the surface of the skin, keratinized cells. Again, waterproof, and then also hair has a degree of pigmentation to it. So that means that there are melanocytes as well that are going to contribute to the final appearance of the hair. Now remember, as we look here at the model or the picture, that any part of the hair that's above the surface is called the shaft. So the visible part, anything below the surface is called the hair root, and the hair root is physically enclosed in the hair follicle. And the hair follicle itself is derived from the epidermis, and also, even though it's never really apparent, from the surrounding connective tissue. But you can see this basal layer basically kind of extending down along with the spinosum, and anyway, that forms the wall.